Um, Good morning. Can you all hear me okay? All right, so welcome to our Everglades REC faculty programmatic seminar series. It's a group of seminars that provide an opportunity for faculty members to give an overview of their program. So potentially a little bit less formal than a traditional seminar. So satisfying professional curiosity, but also hopefully promoting exchange of ideas and maybe collaborations. For those of you who don't know me, my name is Julien Beuzelin. I'm a research and extension entomologist here. And so today we'll talk about my research and extension program in entomology at the Everglades Research and Education Center, Everglades REC. But before I go further, if the computer wakes up, I don't want to. All right. I would like to mention the names of the students who have contributed to the studies. I'll talk about Eric, Wilfred, the second Eric, Larissa, Carol, and Bryn. Bryn currently is my only student. She's working on her PhD, but Carol is going to come back for her PhD later this fall. I would like also to mention 3 extension agents that have been major contributors to the studies I'll talk about today. Matt Van Wilden, Stuart Swanson, and Anna Mesaros. Just in case you're not familiar with the Everglades Agricultural Area, or EAA, this is this area here, about 700,000 acres of land south of Lake Okeechobee. And it's characterized by soils with a high organic matter content. But often when we talk about the EAA, or at least I know for myself, I often include also the C-139 basin and some of the areas surrounding here and here, which actually have sandy soils. So and you can see that actually in that particular area, that aerial image, you see the numerous fields. So it's really dedicated to agriculture. And the yellow star is where the Everglades REC is. So in this area, crops are produced on about 500,000 acres for a value annually that exceeds $1 billion. And sugarcane is the major player, grown on about 400,000 acres. But you also have rice, sweet corn, leafy vegetables, snap beans, peppers, and so on. And actually in my program, we've had at least one project on each of those experiments. So sometimes a sugarcane stalk will have that type of damage. Or a sweet corn ear will look like this with a big fat worm, or a celery stalk will be damaged. And in a nutshell, my job is to prevent this from happening. And to do that, so we develop and refine management tactics for insect pests. And in my program, we have 4 main components. First, stewardship and diversification of insecticide use, ecology of sugarcane and rice insects, sampling and landscape ecology of corn seal flies, and we also focus on response to the threat of emerging and invasive pests. So it's not a spoiler here. I will go through those 4 points and provide some details about what we do. And so first, I will talk about insecticide. We work a lot with insecticides. And I'll start with those guys. Do you have any idea what that is? It's a worm, yes. Good job, armyworm. Yeah, these are fall armyworms, and actually they are one of the most serious problems we have in sweet corn. And every year we have 1 to 5 insecticide efficacy evaluations in the field at the research station, where we evaluate both experimental and commercial insecticides for efficacy against fall armyworms. And for example, our latest evaluation, we looked at different volumes of applications for an insecticide to see if we needed to revise the label. So this is the kind of work that we do, for example. Still sweet corn, but those are a little bit different. Any idea what kind of insect? So these are maggots. So these maggots feeding on the ears are the larvae of what we call corn seal flies. And these are the most damaging pests that we have in sweet corn in Florida, because you definitely do not want to have maggots like this in the ears. And so, say? Yeah, well, these are not their eyes. Do you know that this is their rear end and this is how they breathe? Fun fact. Let's move on. All right. So actually, these are the larvae of 3 species. And so these are the adults of those 3 species. So you have Euxesta stigmatias here, Euxesta eluta, and Chytopsis macilla. And there are some morphological differences. You see the bands on the wings are different. And you'll see later on in the presentation, there are also additional differences. So to manage corn seal flies, our grower primarily rely on frequent applications of pyrethroid insecticides. And we often add PBO, which is a synergist, to increase the efficacy, or we tank mix with methamil, which is a carbamate insecticide, again, to increase the efficacy. And in spite of that very intensive regime, management failures can occur. So is it possible that seal flies have developed resistance to pyrethroids? That could explain why the pyrethroids are not working very well. So this was one of the objectives of the second Eric for his master's degree. He developed a glass vial bioassays to determine pyrethroid susceptibility in corn seal fly populations collected in the area. And so this is how these bioassays were conducted. We used scintillation vials. We put insecticide solution in there, roll them on a hot dog roller to have an even coat. And then we took flies, we anesthetized them with CO2, and then we put them in the vials with different concentrations for 24 hours, and then Eric would rate whether they were dead or alive. So we would see how they would respond to the pyrethroid. And so these are the results that we had. And so we had 1 Euxesta eluta population from the lab, 2 additional Euxesta eluta populations from the field, and all the others were Euxesta stigmatias populations. If we compare the lab population to the field populations, you can see that the lab population was at least 8.2 times more susceptible. And if we look at this one, it was that lab population was 265 times more susceptible. So there was a huge discrepancy between the lab population and the field population. Also, what was really interesting is if we compared the Euxesta eluta populations versus the Euxesta stigmatias populations, overall, the Euxesta eluta populations were more susceptible to the pyrethroids than Euxesta stigmatias. And if we focus on just Euxesta stigmatias, and now we compare the populations from non-treated fields versus populations that were collected in treated fields, you see that difference. in susceptibility, clearly indicating that where we had selection pressure in the commercial fields, those flies were less susceptible. So an indication at least of early development of resistance. So if we have resistance to one type of insecticide, what do we do? Well, we use another kind of insecticide. And so the other component of Eric's project was to look and try to identify potential alternatives to pyrethroids. So we worked on our Euxest diluta lab colony, and we had 20 commercial insecticides. So they were all commercial, but some of them were registered, some of them were not. And the first experiments we did, these were topical assays. So we used Dr. Odero's spray booth. So it can spray the insecticide in that little booth, so it somewhat mimics a sprayer. And so we sprayed the flies with the different insecticides, and we checked for mortality after 24 hours. But we were also interested at looking at the effect of ingestion, because we knew that the exposure could really change the activity. So Eric developed that assay where he put those micropipettes in the vials, and that would force the flies to feed on an insecticide solution. So we were able to look at the effect of diluted insecticides, but when they were ingested. So these are the results of the topical exposure assays. So it's percent mortality here. And you see that these insecticides worked well. They were actually all pyrethroids. We had some effect of spinetoram, that's radiant, and everything else, nothing worked. This is the control here. What was interesting is when we looked at ingestion exposure, this is when it was interesting, because now you see spinetoram has nearly 100% mortality. But here you have 3 neonicotinoids that are also providing good control. So this experiment showed that, unfortunately, right now, the pyrethroids remain the only viable option for the foliar applications that target the adults, like what we do. But we have insecticides like spinetoram or some neonicotinoids that can be highly effective if they are ingested. So one of our next steps will be to see if we can develop baits that force the flies to feed on them, and also work a little bit on concentration. Another one for you, what is this? Good job, Hardev, thank you. It is a wire worm, but then what is this? All right, so Hardev was right. This is the wire worm, and this is the larva of a click beetle. And the most common, so the wire worms, they live in the soil, and the most common species we have here is Melanotus communis. And this is a major pest of sugarcane, newly planted sugarcane, because it feeds on the buds of those seed pieces and also on the roots and shoots of the young plants. So it results in stem loss. Sorry, 2 slides were swapped. To manage wireworms, flooding can be effective for several weeks. It can potentially grow flooded rice, but this is, that's interesting. Okay, but this is not always practical. You can also apply at planting an insecticide for it. The trade name is Thimet, and Thimet is effective. However, it's highly hazardous, and on top of that, it's somewhat expensive. So there are 2 companies that were interested in obtaining registrations for these 2 products, Platinum and Nerisma, for wireworm control. And so with these particular companies, we work to try to identify the best way to use these products for wireworm controls. And so we conducted lab bioassays. So again, in the spray booth, we would put field collected wireworms individually in those cups, and spray them, give them a little bit of food, and see what mortality is. Or we can do outdoor experiments in large containers. The good thing is here, you do have sugarcane, and we infest with live wireworms, so we can see the injury on the crop and the effect on the wireworms. And we've also done on-farm trials in cooperation with Florida Crystals and US Sugar. They leave the furrows open with the sugarcane. We apply the insecticides, and they come and cover, and we can assess how the insecticides have worked. So for example, here, this is the results of one of the latest field trials where we looked at Nurisma at 3 rates, Nurisma and the fungicide, platinum, thimet, and we had non-treated check. I'm reporting the number of shoots and tillers per 30 feet, 4, 7, 10, and 13 weeks after planting. Across the observation dates, there was a trend for differences among treatments, but 13 weeks after treatment, after planting, there was a clear pattern here that was significant, with the lowest stand or plant population for the non-treated check, intermediate group here with the insecticides, and about 50% more sugarcane plants when you had the insecticide and the fungicide. So these insecticides are very promising, and for platinum, we are waiting to hear from the EPA anytime now if we have a registration. What is interesting is our studies and other studies in other systems show that actually platinum does not kill the wireworms, it just incapacitates them. So we don't know what the long-term effect of the insecticide will be. For Nurisma, what's happened is the company has decided to pause indefinitely the registration effort. So we don't think we're going to get it, but at least we have generated data. And if the regulatory process moves forward, we will have data to support the use. So all this work on insecticide really helps us identify experimental and commercial insecticides that are effective against pests. So this work supports the registration efforts for the insecticides, but also all our recommendations. Also, we can determine if insecticide resistance contribute to control failures, like I showed for the corn seal flies, and actually we've done similar work for armyworms. I did not have time to show that here, but we work also a lot with biological insecticides, softer on the environment. They fit also organic systems, and so that was one of the focus of Wilfred's masters on sugarcane aphid. And one thing that I wanted to mention that I think is very important is throughout this whole process, our stakeholders, the growers, consultants, and extension agents are involved. So we exchange information, and so especially with the on-farm trials, so they're aware of the research findings. So that was the first component. Now I'm going to move on to the second component of my entomology program. Insecticides are important, but understanding the ecology of insect pests is equally important because it can tell us if and how modifying the environment and the interactions that the pests have, if that can be used to lower the populations of those insect pests. So here I will not ask a question. This is a sugarcane borer. As the name indicates, it's a pest of sugarcane. And sugarcane borer populations have drastically declined since the 1990s when a US sugar released for about 10 years parasitic wasps to control this insect. But actually, when started the job in late 2016, we had questions about the pest status of the sugarcane borer and some of those wasps. So at the time, Eric Roldan, who was my first student, conducted that large experiment where we had 32 fields throughout the area that we monitored for 2 summers. And those fields were stratified so that we had a shallow muck, deep muck, and sandy soils. We looked for sugarcane borers, and the good news is that the populations were very low. We also took the opportunity to look at ants because we know that ants are very important predators of sugarcane borer. And the vast majority of the ants were red imported fire ants. We also had other ants. I believe most of them, Matt, are invasives, by the way, which is interesting. The soil did not impact much the ants, but we still saw a few differences. For example, in sand, there is more, you have more crazy ants. So we were able to see that we had a robust population of predators, and also the very few sugarcane borers that were recovered. If this one, for example, you open it up, you see it had those little, look like rice kernels, or this one, you see what emerged, there is a maggot. Actually, these are the larvae of wasps. Wasps that lay their eggs in the larva and eventually kill it. And so actually we had relatively high parasitism rates telling us that those natural enemies were doing their jobs. So what's going on in rice then? So the borers can affect rice, but sting bugs are the main pests. There are two main species in Florida rice, Ebolus pugnax and Ebolus insularis. And we are the only rice industry in the continental US that has Ebolus insularis. So that makes us special. And so whatever we do is somewhat more interesting than in other states because we have that pest complex. So what we wanted to do, because natural enemies can control insects, was to identify rice sting bug parasitoids and quantify parasitism rates. So that was Carol's master's project. And again, it was a large on-farm study over 2 summers. Each year, we had 27 commercial fields. And for this, we specifically focused on eggs. So here you see this is Carol, and I'm not sure if this is somebody helping Carol in the field. We use these quadrats to have our sampling area and sample the plants for naturally deposited eggs that we collect. But we also use what we call sentinel eggs. So we bring sting bugs in the lab. We let them lay eggs, and then we take these eggs and we, well, Carol glues them on waterproof paper, and then we can deploy them in the field. So it's a more controlled way. We know where they are. And for example, we did inside the field versus the field margin, and we did the 2 species, for example. And then we leave the eggs for 48 hours. And whether it's the sentinel eggs and the naturally deposited eggs, when we bring them back to the lab, sometimes this is what happens. This is what comes out of the eggs. So again, these are wasps that actually lay their eggs in the sting bug eggs, killing the sting bug. Pretty cool. So these are our results. Forgot to mention, we also stratified so that we would do conventional versus organic. We thought that would be an interesting comparison. So here it's for the naturally deposited eggs. We look, well, a few things, number of eggs, but percent parasitism and percent wasp emergence from those parasitized eggs. We did not see differences between conventional and organic, but you can see that we had at least 60% of the eggs that were parasitized. And from those parasitized eggs, at least 50% of them produced new parasitoids. We collected 4 different species that Carol identified, and 92% of them were that particular species, Telenomus podici. For the sentinel eggs, it was somewhat the same pattern, no difference between conventional and organic, but overall, the rates were lower, indicating that the parasitoids did not like those eggs as much, or that there was some problem, but 100% of the wasps were Telenomus podici. So, you know, they are very small, so we took some pictures. So these are those wasps that lay their eggs in the stingbug eggs, thus killing the stingbugs. So parasitism by Telenomus podiaceae was relatively high, and Telenomus podiaceae has been produced commercially. So a biological control program relying on the release of those wasps to increase the stingbug egg parasitism, which would then decrease the stingbug populations, might be a strategy that we would want to explore to see if it can go away from insecticides under some conditions. So that was it for the component on sugarcane and rice insect ecology. All that work allows us to determine the relationship between the insect pest and their environment, and especially with what we've done there, the natural enemies, the predators, the parasitic wasps. And as you can see, that work really emphasized the value of biological control. And especially in sugarcane, it's really key to avoid disruption of those natural enemies because the sugarcane borer populations would go up. But also in rice, we could potentially consider releasing those parasitoids. The other thing that I would want to mention is, like for the previous component, some of the stakeholders are key partners. And so here you see Matt Van Wilden checking some cane. And so Matt participated on the committees of two of the students involved in those studies. And again, working with Matt has allowed us to do extension while we're doing research at the same time. So if you remember, I had mentioned that seal flies were our major problem in sweet corn. And I also mentioned that ecology was very important. So the third aspect of what we do focuses on sampling and landscape ecology of seal flies. The first thing that I did when I started here was to try to develop a trap that we could use to monitor corn seal flies. The graduate student working with my predecessor, Greg Nestle, had done a good job starting that work, so I could build on that. But we tested numerous traps and numerous lures, and we found that those multi-lure traps, so they have that transparent dome and that inverted yellow funnel here, baited with ammonium acetate and 1,4-dimethoxybenzene. The dimethoxybenzene is a floral volatile. These traps were attractive to the three corn seal fly species. And so if you open those traps, you can see often you'll have seal flies in there. And here you see this is Larissa checking for corn seal flies in the field. So if you remember earlier, I mentioned that we, in spite of the very intensive insecticide management, we had failures sometimes. And another insecticide resistance is potentially a factor, but another factor is potentially the adult movement back into the fields after treatment. And Gaurav Goyal, he was Greg Nestle's student, started describing that, but it had really not been done in depth. So we wanted to resume some of that work. And so part of Larissa's, and if you start seeing a theme with maps, we like to drive around, Larissa's project, we focused mostly on the EAA, but we also had sites in Homestead. So we had 2 years and 55 commercial farm sites. Did not check them all at the same time because we had sampling in the winter, in the spring, and in the fall. And so our first step was to look at adult abundance, relative abundance in the corn versus weedy areas around the field, and also sugarcane for the EAA. For Homestead, we looked at tree nurseries. So here you see Larissa's results. For the spring, this is when the seal flies are the worst. We are reporting the number of adult seal flies per day per trap. And you can see that relatively consistently numbers are low in corn. This is normal because corn is sprayed with insecticides intensively. But we have substantial numbers. in the sugarcane and in the weeds. Also, if you look at species composition, there are some differences I will not focus on, but relatively speaking, we have a little bit more Euxesta stigmatias in the corn than in the weeds. So that was on commercial farms, but we took the opportunity of having a research and education center here. So we had a grid of 150 traps, and we also mapped seal fly populations here at the center. And this is for Euxesta eluta. And you can clearly see that you have areas of concentration. And for Euxesta eluta, it was either non-treated corn or some tall sugarcane. Now, what I want you to focus is this is Euxesta eluta, and we're going to shift to Euxesta stigmatias at exactly the same time. And you see those patterns are very different. And Euxesta stigmatias was a little bit everywhere, but primarily associated with corn, whether it was sprayed or not, so showing a strong association. So those species are different. So this work on sampling and landscape ecology of seal flies really helps us monitor the fly populations with the traps, which can provide information on the adult distribution in the landscape on commercial and research farms. So that's good. We've highlighted differences among species, and so we were able to put numbers on things we thought were happening, and it's become clear that a new management strategy, including those habitats surrounding sweet corn fields, would be welcome. So actually, to understand those interactions, characterizing the movement of the adult flies and potentially manipulating the movement of those flies would be critical. And this is part of a project that we have with Marcio Resende. And this is actually the focus of Brain's PhD. So she's going to talk a little bit about what she's been doing.
Good. Awesome. Okay. So as we just talked about, understanding the movement of the adults and our potential ability to manipulate that movement is very, very important for corn silk fly control. Now, in the literature so far, it does not exist for a movement strategy to understand the movement of corn silk flies. But here's the good news. There is literature on flies in general. And for each of these individual studies, the vast majority of these studies focus on something called a mark, release, recapture method. Now, I'm going to talk about that method in 1 second, but the first part of that method is marking. So the first thing I have to do is figure out how to mark these flies. Now, when I was doing this, I was interested in 3 methods that were most consistent or commonly seen in the literature. So the first one was something that we called pupil marking, which you can see all the way up at the top. It's just the heads. The second one was called external adult marking, which is the second one in the middle, where you can see that one fly is neon blue. And the final one, which we called internal adult feeding, and it's a little difficult to see, but the gut here is a little bit red. So that's what we worked on. Now, our objectives for this study were 2 main things. First thing, how much dye do I need to apply to make it blue? That's an important characteristic, right? And if I'm making it blue, am I impacting its behavior? Because if a fly can't fly, that's not a very useful study. Now, almost more importantly, I need to make sure if you're blue, are you going to die? Because dead flies don't fly very well. It's kind of hard to study their movement. Last but certainly not least, I need to make sure if I make it blue in the lab and then let it go into the environment, how long is it going to be blue? By the time I'm looking to recollect it, is it still going to be blue? So I'm interested in the rate of degradation. Now, the results so far, this is the only part that I have results on, but we've found that if we had to make a suggestion for using a marking method for a mark, release, recapture study, we would recommend using external marking. As you can see, it's pretty clear if it's blue or black. Second picture. And in addition to this, at moderate to high concentrations, we got 100% marking. And for each of these 3 methods, behavior was not significantly impacted. So that was a great result. Now, with this information, once again, the whole point of marking them is so we can release them and catch them again. The reason that we want to do that is because that gives us important information on how they're moving in an environment naturally. So our goal is to identify 2 things. First of all, If a corn silkfly is not in a sweet corn field, where is it? And if it's in that habitat, how long does it take it to get back into the sweet corn? That's really important. So we're interested in doing 6 different releases. 3 of those releases are going to be at EREC. You guys can come see if you want. And the other 3 are going to be at commercial farms. So what we're going to do is that for each individual site, we have 4 release points, which means 4 places that we're actually releasing the flies. So we're going to release them from the north of the field, east, south, and west. And ideally, each of those directions is going to correspond with some type of habitat. So let's argue, for the sake of argument, the north of your field is actually a sugarcane plot, but your field is sweet corn. So our goal is that if we are releasing a bunch of flies from the north and we're seeing it in our traps, it's good to assume that they're probably coming from sugarcane, right? So now I can say, hey, grower, if you're seeing corn silk flies not in your field, but you're still seeing them in your traps, maybe they're coming from sugarcane, you should probably spray there. Now, what's more important is they can say, okay, great, they're in the sugarcane, I don't care. How long does it take for them to get in my field? I want to answer that question. So depending on how frequently I check my traps, every hour, every day, every couple days, I can be able to tell the grower, hey, listen, I released these flies in the north and I caught them in my central trap an hour after I released them. So I can safely say that if my trap is 30 meters away from that release point, these flies can travel 30 meters an hour. It's incredibly fast. So now I can go to the grower and I can say, hey, listen, I can tell you what habitat to spray and I can tell you when to spray it. Now, that's how I can understand their movement in a natural environment, but I want to go a little bit further. I want to be able to tell the grower, hey, whoa, I can tell you where to spray, but what if I can do you one better? What if instead I can tell you that they won't get to your field at all? If I can manipulate their movement, I can tell a grower that, and I can manipulate their movement in 2 ways. I can attract them to something else, or I can repel them from the sweet corn altogether. So my goal for this study is to identify what compounds are attractive, what compounds are repellent, and at what concentration should I apply those attractants or repellents. Too much of a good thing can be a bad thing, right? Have you ever been next to someone who's wearing a lot of perfume? Now, the significance of this study is I can say, hey, whoa, listen, you know how they move in a field, and that's fantastic. But if they don't get there at all, it's even better. So I can do this using 3 potential methods. 2 of them are in the lab, and 1 of them is in the field. 2 are in the lab because it's easier. So the first one's an olfactometer assay, and the second one is a 2-choice cage study. The final one is obviously just spraying in the field and seeing what happens. Now, last but certainly not least, and I'm very excited for this one, we're interested in employing a potential biocontrol method. So it's one thing to go to a grower and say, hey, we can possibly get them away from your field, but it's another thing to say, if they're in your field and your insecticides aren't working, I have another alternative for you. So in this method, what I'm aiming to do is use something called an entomopathogenic nematode. It's a nematode that affects insects. So I want to be able to go to a grower and say, hey, listen, I've identified a species where if you deploy this in your field, it can control corn silk flies when they're in their immature stage. That's awesome, right? So I can do this in 2 assays. First of all, I want to see if it works in a lab. That's the first thing I'm going to test. The second thing is if it's employable in a field. If a grower can't use it, it's not super helpful. But the significance of all of these parts of my dissertation is to be able to present growers with information on how to control the pest pressure in their fields. How are they moving in your field currently? How can I get them out of your field? And if I can't get them out of your field, how can I control them since you are seeing management failures? Ideally, we can start to reduce our use of insecticides, which is healthier for the consumer, for the grower, and for the environment. That being said, if you have any questions, you can see me afterwards, but I'll turn it back over to Dr. Beuzelin.
All right, thank you. So it looks like we're about to solve the sealfly problem, so we'll move on and talk about invasive species or emerging and invasive species. And we have about a little bit less than 10 minutes, I think, before we wrap up. But I like to, I had more examples, but talk about 4 examples. That's the last component of our program. And 4 examples, the first one is the Mexican rice borer. So that's one insect that concerns us because it occurs in Texas and in Louisiana. And it was first detected in Central Florida in 2012. We've collected some insects to do some sequencing to figure out where they're coming from, but they are still in the freezer, so more to come on this. But this is a pest of sugarcane and rice. So we need to be proactive. We are lucky that for the Mexican rice borer, a synthetic sex pheromone has been developed. And so if you put the sex pheromone lure in one of those traps, so the lure is here, you see males think it's female, they'll come and eventually they will drop in the bucket and you can detect the moths, and this is a good detection method. So every year in the EAA, we've deployed 16 to 30 traps, averaging 20. So for example, these are the traps that were deployed in 2022. And usually Donna is the one checking those traps once every other week or once, yeah. And so far, the great news is we feel pretty confident that this insect is not here. But we've also sampled in central Florida. So these are the traps that we had in 2023. And again, Donna was the one in charge of that. So we had 34 traps in an area where we expected this to be about the southern edge of the invasion. And you see those traps are the positive traps and the yellow traps are the negative traps. But we had some new detections here. And where is the other one? Yeah, here. Indicating that actually the insect is moving slow, about 2.5, 3 miles a year towards the sugarcane area. But if it's not intentionally introduced, it will take a while to get here, but it's moving. So we keep monitoring to ensure that populations are detected if they move quickly. The second example is different. Delphacids. There are 3 Delphacids. These are plant hoppers, the small brown plant hopper, and white-back plant hopper. These invasive insects, if they become established in our rice industry, they could be game changers. And on top of that, they are vectors of diseases. So we've been monitoring for those insects. As far as we know, where they are not here. If you wonder what that big tube by the weather station, what that is, this is a suction trap. So it's a big PVC pipe with a motor and a fan. So it sucks the insects that are flying above. And we've been continually running that trap since August 2019. And we collaborate with FDAX DPI to identify the insects. We also complement some of that work with field surveys. So here you see this is Amir and Donna about to go sweep the rice fields for the Delphacids. And actually, when they were sweeping, they didn't find an invasive plant hopper, but they had the first continental record for leaf hopper that previously had only been described from the Seychelles and been intercepted in shipments from Colombia. But it showed up in our rice fields. We don't think it's going to be a problem, but it shows that anything can show up in our fields at any time, and we have to be extremely vigilant. That's one thing. The other thing is this insect, the expert at the Everglades REC is Dr. Defenmu. So she's taken over the project, and I will not be working anymore with Delphacids. We found this one, we're moving on. Third concern, any idea what we're looking at? Peppers. All right, pepper that had an issue with an invasive species new to Florida. Do you see those little insects on the flower? Any idea? Good job. Yep. This is Thrips parvispinus. This is the first detection in North America was in 2020 in Florida. And it was observed for the first time in Palm Beach County vegetables in 2022. And so it's really a problem in peppers, but also infests things like cucumber. And so as you can see here, actually, so this is Defen and Anna working together on a cucumber trial. And so we've been working together trying to address the situation to provide growers with management solutions. And the last one, because I think it's really interesting, was the situation in parsley and celery was brought to my attention and Anna's attention fall 2021. Some tunneling like that, like you can see in celery. You see the larva here, some stunting in parsley. And we had no idea what that was. It looked like carrot weevil, but we're not sure. So we reared those larvae and to adulthood, and we had those little weevils. So we sent them to FDACS. They were not sure what they were, so they sent them to one of the leading world experts in Canada. And these insects are Listronotus sparsus. And this is a relatively widespread weevil species that had never been reported as a pest. But for some reason, we're not sure why in the AA it became a problem. So with Anna, we've worked together on this too, learning a little bit about the biology, how quickly it impacts the plants, the time it takes for a life cycle to complete and so on. But also going back to insecticides, especially in vegetables, it's very important. We've looked at products that could be used. And I know we often say that a picture is worth 1,000 words, but this was parsley here. And you see that bed was not protected. And this one was protected with insecticides. And you clearly see the impact of the weevil. So we're able to learn about the weevil, but also learn about what could potentially be done to control them. So to conclude on that fourth component about emerging and invasive species, that work really shows us that we have to be proactive because we really want to prevent the buildup of pest populations without us being aware of that. And it's happened in other systems. And that's really the example of Mexican rice borer and the delphacids. But we also have other problems that are already here, and we have to provide emergency recommendations. So this is the kind of work that we do. But as is the case for the other components, everything we do, we involve the stakeholders, whether it's the extension agents, the growers, the consultants, the agrochemical company reps, because especially for invasive species, we are all first detectors. What that means, we can all come across something that is new that we need to report and take action. So education is really, really critical. And so speaking of education, I have one last slide about extension. I will not expand much on extension because a large portion of what we do, our research and extension programs are really integrated. It's a continuum. There's no separation. But we do the traditional extension methods, especially with some of the meetings we have here and the field days. So that complements all the on-farm work that we do. But we feel that with the clientele we have working directly with the growers, the consultants, and the agents, it really goes a long way. Before I finish, I have a very long list. I did not put the logos, but wanted to thank again the graduate students. We all know how important the work done by the graduate students is. The extension agents I mentioned before. Technical assistance. Donna has been my right arm for 6, 7, long time. Doing all of that, Amir and some interns, including Hayden with us today, all the crop consultants that I have helped, the growers, you know them, these are all the major growers here. And how all of this is funded, we have good partnerships with agrochemical companies, but also support from the growers, FDACS, some from IFAS, SARE, APHIS, NIFA. All of this comes together so that we can do that work. So hopefully that gives you an overview of what we do. Obviously, I could not cover everything, but if you have questions for me, Bryn, Anna, Donna, Matt, please let us know. We're here. Eating pizza at some point. Yeah, if it's about trapping, ask Donna. So thank you very much. Yes. I can, you sure? Let me run. I'll do that.
Now, yeah. Uh, Great program. My question is related to, you're monitoring these insects, right? Is there any tendency you see with how the weather is changing? And if you believe we're gonna get, newer insect pests and
So,
how that's gonna go? What's your opinion on that?
so I think, well, the primary factor is trade and movement of people. People bringing what they should not bring in their luggage. So this is primary factor, but obviously, especially from tropical areas, things that would not have become established in the past can potentially become established. So yes, we do expect some northern ranges to expand. So with increased human activity and climate change, the issue of invasive species is expected to become worse.
I have a question. So as we know, for the sugarcane planting, we have Florida and also Louisiana. They're also planting the sugarcane. So compared with Louisiana, do we have more insects, like both in the species, also the density? And what kind of reason makes that?
All right, how long do you have? All right. My knowledge of Louisiana is a little old, but so soil insects definitely worse in general in Florida, especially in the organic soils with the wireworms. They are definitely not as bad in Louisiana, whereas in Florida in organic soils, the wireworms are terrible. White grubs in Louisiana, I don't know. Here, they are not much of a problem, but I don't know if you knew Dr. Cherry, who retired a year or two ago. He had worked a lot on white grubs. These were problems here, and I believe they were not problems in Louisiana either. So soil insects, definitely Florida, more job security. Above ground insects, so limit the soil line, you have lesser cornstalk borer, and this is more a problem here. So that's a caterpillar. It feeds on young plants and mat. I don't think, I mean, once in a while they can pop in Louisiana, but I've seen them in sorghum over there, but yeah. So here we're doing better than Louisiana. But then if you go a little bit further above, in Louisiana, they have issues with sugarcane borer and Mexican rice borer, and right now we don't. And they also have some issues with some piercing sucking insects. So one Delphacid penthopper, so-called West Indian cane flies, cause some issues. And there are some flare-ups of aphids, which we've had in the past here, but it seemed it was more associated with some varieties, some issues with mites too. But so I would say that overall below ground, we have more insect issues, but above ground, they have more insect issues. Yeah, my son. Oh, sorry.
Well, thank you. Thank you so much. Great program, excellent presentation.
Thanks.
You mentioned a couple of times that soil depth, shallow, deep, and sandy soil. And you mentioned also the ant population, different soils. So do you have any a short list of mechanisms that these different soil types and properties affect these insect damage and population?
The hypothesis, so let's consider muck with the bedrock and muck, shallow or deep. And the idea was the ants nesting in the soil. When you have less soil, they have less space. That's one thing. But also we know that, well, shallow muck dries quicker, but floods quicker. So it's more often disrupted or could favor insects that are adapted to flooding, for example. And red imported ferret is very well adapted to flooding. So we thought that could be a factor. And actually we saw a little bit more ferrets in shallow muck. So these were some of the mechanisms we thought could play a role.
This was important for the ants, or was it anything? Did you find that important soil type in any other of the research you did?
So we only focused on the ants and the borers for that, yeah.
Thanks.
So Julien, what does it take to have a biological agent like a parasite to be approved? Is there like a regulatory mechanism, or if it works, you just can't release it?
I do not know very well. If you have to import a non-native species in the environment, say, for example, it is determined that a wasp from somewhere in Southeast Asia would be a very good candidate. It does not occur here. There would be a lot of work that would be needed. By the way, 100 years ago, you just needed to bring it in your pocket. So things have changed a lot because it's important to be sure that it has narrow spectrum and so on. What we're looking at, so for sugarcane borer, the two parasitoids that are established are non-native. One, the solitary one, was introduced from Peru in the 1930s, and Cotesia was introduced by the USDA in 1950s. It was established, but was doing okay. And then US Sugar, they brought in their own, I believe, Brazilian strain, and they released it, and it looked like it did better. So I don't know what the process was. What we're looking at, it's a native parasitoid. And actually, it looks like it's two different species, but it's Carol's PhD. Yes, Larissa?
So you can choose to answer my question based on your own opinion, what growers do, or science that you know. I'm interested to know what would be the population threshold required for application. Would it be a better approach to apply an insecticide while the population is still low, or we have to wait and reach that economic threshold to deploy our applications?
It would be very dependent on the system and data available. We're talking, for example, sugarcane borer. Before, when populations were higher, you had scouts, and it was an empirical threshold. But and I forgot, but I believe when they had 5% infestation, they thought there could be issues, and actually it's the same threshold in Louisiana. But then they would dissect the insect to see if they were parasitized or not. So if they were not parasitized, they would treat, and if they were parasitized above a certain threshold, they would let the parasitoids do their job, and it worked. So some of those things, you have scouting, you reach a threshold, whether it's empirical or it's been backed by pretty comprehensive studies, it can work very well. With the vegetables, you have an aesthetic aspect. There is a lot of money involved, and sometimes that threshold is practically zero. So you typically start with a schedule to keep things clean or almost clean, or you're always very near spraying. And whenever things start becoming a little worse, then you increase your frequency. So well, in my opinion, it's really dependent on your system and the data available. And then also how practical, for example, the soil insects, even if you have the threshold, if you cannot get to them, you have to treat prophylactically.
Oh, I have one last question.
Are we kicked out?
But
I work here, so you can.
Okay. Back when you were testing different insecticides against wireworm on sugarcane, there was a treatment with a fungicide included.
Mm-hmm.
What was the theory or hypothesis behind that treatment?
So we know that issues with fungi by themselves can reduce the plant population by degrading seed cane. But also the wireworms can potentially cue on products of decay in the sugarcane, so you can potentially have an interaction. So we didn't even go there, but the idea was maybe we could get some synergism with the fungicide. And anyway, if you plant mechanically and you're already spraying the insecticide, you'll probably go insecticide-fungicide combo. And that was probably the most likely treatment that have been used in the field.